So we will continue with our eukaryotic gene regulation. And we were learning about several of the things, and mostly we have noticed that there are, like in prokaryotes, there is a great deal of regulation that happens at the level of transcriptional initiation. But otherwise, in eukaryotes, one can regulate, or a gene could be regulated in many, many steps. So let's recollect some of the words like enhancer, that is the activator binding site, and we have Tata binding protein here, TBP, along with TF2D. There is mediator complex, RNA polymerase. We also discussed about phosphorylation of RNA polymerase, how it will allow the binding or formation of an initiation complex, and for it to proceed to transcription elongation, then that needs another level of phosphorylation on the CTD. So we'll just quickly have a look that are variants or are at a higher complex levels in eukaryotes. The first thing obviously is that in eukaryotes you have the whole genome bound by basic proteins with on surfaces are made of large number of basic amino acids, and therefore they have interaction with the DNA, which is on the phosphate backbone at least is negative in nature. So nucleosomes are made up of histones, and histones are non-specific DNA binding proteins. They're not sequence specific. So and when they are bound, binding of DNA around nucleosomes is important for compaction, because a long DNA should be packaged into a very small tiny nucleus. And for that to happen, when compaction happens, there are obviously problems that will associate accessibility of the DNA or the regulatory elements such as promoter or enhancer to the binding proteins, like even mediator complex or RNA polymerase or any initiation factors. So there we need several machinery to unfold these DNA molecules to spread around like this and so on, or at least remodel it so that these regulatory elements become accessible to their respective regulating proteins, right? And for that we will need at least the histone tail modifications or post modifications are important in managing whether they should be compact or should be little bit or loose and so on. And that modification is done by several proteins like histone acetyl transferases or histone methyl transferases, deacetylases, demethylases, and so on. Not just by post-translational modifications, but we can also do by remodeling the nucleosomes itself, what is not accessible, because you can slide the nucleosomes here and there. You can make these genetic elements accessible to initiation factors or regulators. So those are the two major ways. One is through you will achieve the accessibility by post-translational modifications of the histone tails. The other one is by modulating the positioning of nucleosomes within DNA, by changing them here and there. So at least in the epigenetics part, when we say, I think I have to define epigenetics. I will do that I did that before, long time ago, but I will do that again later on. But what we should know is we spoke about transcriptional modifications, post-translational modifications of histone tails. And they can be acetylation and methylation, phosphorylation also there. Mostly acetylation and methylation play a big role. And from changing to very compact heterochromatin to little bit loose leaf organized chromatin, that is the euchromatin, acetylation is involved in that. So acetylation is mediated by histone acetyl transferases, and once acetylated, usually it becomes euchromatin. That means it is spread out a little bit, open and accessible. DNA is accessible, and that leads to mostly a gene expression, gene transcription will happen. The opposite action or antagonistic action to histone acetyl transferases are histone deacetylases. They remove the acetyl groups from the histone tails, because of which the chromatin becomes compact and genes are now kind of silenced, or in because of formation of heterochromatin. Similarly, there is also histone methyl transferases and histone demethylases. These are sets of, again, enzymes that either favor methylation or favor demethylation. Methylation can be, it's can cause gene transcription in some cases, methylation of some residues. Sometimes they may cause gene repression, right? H3Ka, histone H3, K4 is lysine at position 4, and so on. So we have seen several of these examples, and we have also come to learn about several elements that are present. Promoter elements, eukaryotic promoters uh, are large and a little bit spread out, like the initiator region, downstream promoter elements, core promoter elements, upstream promoter elements, and so on. But there are also regions called as enhancer regions, which are actually activator binding proteins, right? When the activator is, these are the activators, they bind to the enhancers, because of which they can influence the transcription of the from the adjacent promoters. And when they are too large, then the distance between the enhancer and promoter is large, the DNA should bend. Only then you will become, the activators will come in close proximity with the promoter regions. So what also may happen, one second, sorry, my bad. Yes, what also may happen is there are other regions called as insulators. Insulators bind to, insulators are regions where certain proteins, something like CTCF, they can bind. We will discuss about this today, and that's one of the important things that you will have to remember. At the end, the genetic organization is important. Say if the insulator is here and not here, and if CTCF binds here, it is not going to have any effect of this enhancer activator interacting with this promoter, right? But again, one enhancer, activators bind to one enhancers could influence two promoters, either say promoter A or promoter B. And which one will which promoter is activated by this particular enhancer is determined by the placement of the insulator, where it is located. So I hope you can see that there is a design, and then based on that, there is the outcomes of the regulation are dependent on that, on the organization. So one promoter could be activated by two enhancers, could be, it depends on the genetic organization. One enhancers could activate either of promoter A or promoter B, or more than one promoter. And whether the promoter along with the activator loops to physically reach a promoter or not is determined by the insulator binding proteins, or CTCF like proteins. We will discuss that. And there are also locus control regions or global control regions, which if these are silenced, this whole locus gets silenced. Those are referred to as locus control regions. So I think I discussed this. Cell is genome is same, but then you end up having so many different cell types, and that means some genes are turned on in skin cells, some genes are turned, and many types of genes are turned off. If you take a genome of a human genome, it has all the genes required to form skin, it has all the genes required to form neurons and red blood cells or bone tissue and everything else. But say, as I said, in this epidermal cells of skin, in this case, the genes responsible for skins or epidermis are expressed, other cell types are repressed. What happens in neurons? In neurons, genes are expressed, others are repressed. And that is how the genetic information or the ability to form any type of cell is present. But by differential gene regulation, that means differential gene expression, we are getting so many different types of cells with diverse functions, right? So I'll just say here, stomach cell, the lining, assume that, which it will it will have, all cells will have general metabolism once. Metabolic genes will be there, they will be expressed in all the genes, and those required for product for having the phenotype of stomach cell will be expressed, and something like neurons, skin, lungs, and everything else will be repressed, and that is how this becomes a stomach cell, right? Just keep that view in your mind. And how is that? We learned also about different levels of genetic organization. Before going in that, I will try to describe what is meant by epigenetics or epigenetic mechanism. Say, for example, you know about -10 region, right? In prokaryotes, it is TAT. I'm just making up so that I'll probably be able to explain what epigenetics means in the simplest forms possible. This is promoter, and you have -10 and -35 and so on. I will give you a scenario where I have so assume that transcription from this gene is going to be, it'll produce 10 mRNA per minute. I'm just making it up. So I'm making another something seemingly -10, but it's not optimal, it is suboptimal. Assume there is because of some mutation or something happened, and you have the same gene, except there is some modification in the -10 region, because of which it is only producing 1 mRNA per minute. Has there been change in the gene expression? Yes. One is producing 10 and the other one is producing only 1 mRNA per minute. The expression or transcription has reduced drastically. And why is this? Why is the transcription different? The cause is in the sequence. The cause is change in sequence. Change in sequence is mutation, right? That is what we studied, right? Any consequence which is because of a change in the nucleotide sequence is a mutation, and it's genetics, right? I will now try to have a little bit different view. As I said, we have some mechanism because of, say, in eukaryotes, we have nucleosomes, and I will say about the same gene, okay? I'm talking about some gene XYZ, and in one cell type, it is very compact like this with nucleosomes, and therefore there is no expression. Assume that there is a gene here which is XYZ. And the same gene is also present on this one, XYZ. There is no expression in the case A, and in case B, there is expression. Has there been a difference in the level of expression? Yes, because here there are 0 transcript or mRNAs that are produced, whereas in the other case you have 10 mRNAs produced, right? And what is the cause for this difference? Is the difference because of change in nucleotide sequence? No, it's not. We didn't change any nucleotide sequence. But yet there is a change in the or difference in the expression level. And that is because of the nucleosomes that are present. And why are these nucleosomes spread out like this? Probably the histone tails here are acetylated. And whereas here probably they are methylated or maybe just deacetylated. Right? So I hope we are getting to understand what epigenetics means. Any change in any change in gene expression that is not because of change in nucleotide sequence, right? Say for example here, if you're methylating a DNA, is it a change in sequence or not? It is not a change in sequence, because say something like CGA assume that there are 2 sequences like this, CGA, one of it is methylated. I'm just representing it as that, as the cytosins are methylated. The sequence is still same, right? But there is, because of methylation, there are consequences on gene expression, which we will see shortly. So this is a mechanism of this is an epigenetic mechanism. Say, and as I was describing histone acetylation or methylation, this is the example that I was telling you. Is there a change in nucleotide sequence? There is no nucleotide change, but there is a change in the modification of histone tails, which is resulting in the expression of genes, and that also will come under epigenetic mechanisms. Chromatin organization, like we will see X chromosome inactivation, and the whole chromosome itself gets inactivated, and that we'll study in stem cell biology if you take it. But otherwise, yeah, you might already have known it. There is something called bar body. It happens one X chromosome in females, where females have 2 X chromosomes, whereas males have 1 X and 1 Y. This is called as dosage compensation, where you only need to have 1 X chromosome active in the sense available for expression. We don't want both of them. It's not good for the cell. So in all of the somatic cells in females, one of the X chromosomes becomes a small, tiny, it's completely silenced. And that it is called as then bar body, the silenced one, right? Only 1 X chromosome is available for expression. In males, there is no issue, because there is only 1 X chromosome, and Y doesn't have many things at all. Very few things are there from Y. But I hope you understood that. And nuclear architecture is the gene. There are certain areas within the nucleus where transcription is highly active. If the genes are brought into the areas of high transcriptional activity, those that need to be expressed, they all are concentrated from different chromosomes onto one or several areas of transcriptional zones. So that way, if a gene is not within the transcriptionally active zones of nucleus, then it is silenced. Whereas if it is in that area, it is usually expressed. So nuclear architecture can also have a role. It's an epigenetic mechanism. So what is epigenetic mechanism? Epi means above, right? Epi means over or above. You can say genetic here is about genes or nucleotide sequence, right? A modification or regulatory mechanisms which involve a modification over the nucleotide sequence, or you can otherwise think of it as regulatory mechanisms that do not involve change in nucleotide sequence. Those all those mechanisms fall under epigenetic mechanisms. In eukaryotes especially, this is the is a very hot topic in terms of research, because if I will even if you don't have to take my course, but there is a fantastic course on Coursera about epigenetic mechanisms. If you ever get an opportunity, and if you read the textbook well now, you have a good foundation to go and start that. There are many more amazing things that we didn't know before, so try to have a look at those. So DNA methylation happens, usually they are called as CpG islands. There are several regions where they have CG like this, which are usually upstream of several genes. Near the promoter or regulatory elements, you have CG. Those are referred to as CpG islands. P is for phosphate between these two, C and G. So there are about 50,000 CpG islands in our human genome, and they can be associated with several types of genes, cell type specific or housekeeping genes. Housekeeping genes are those that are typically involved in metabolism and also building something like general cellular materials. Like you want to make every cell has endoplasmic reticulum, every cell wants to have as a nuclear membrane, Golgi complex, and all those things. So to keep their shapes are different, which will have a specialized one, but otherwise you want actin filaments, microtubules, and several other things. Typically those that are involved in metabolism and those that are involved in general architecture and functioning of basic functions of a cell, they come under housekeeping genes. Cell type specific genes are those, like I said before, those that should be an epidermal cell, then only those will be expressed, but not the neuronal type, and so on. So in CpG islands, the cytosine is methylated on the fifth carbon that we have already discussed yesterday, and then it is now 5-methyl cytosine. And demethylation would have resulted in cytosine. What would happen if it is deaminated? I think it should be here, right? So this is also a question or important for you to think of. CpG islands are more prone to mutations, or they are hotspots for mutations. And why is that? It comes into this. So cytosine, and we are saying that DNA can get methylated. We will see the consequences of this methylation at a later stage. But just we're trying to understand the important the chemical perspective, and also the um, importance of this methylation in terms of mutations. So methylation, and this is the fifth carbon now. You will count it as 1, 2, 3, 4, 5, and 6. So this methyl group is added, and now cytosine has become 5-methyl group. If deamination happens, that means the amino group is lost and replaced with a keto group. What you now have is thymine. Think of it like this. You have, if there is A, T, G, C, A, C, T. Okay, this is a sequence, and this is methylated. Okay, and assume that this sequence has changed, or it got the thymines got demethylated, or what would have happened if thymine gets demethylated? In this case, it would have become A G U C A C U. In one of the assignments I gave you, in a mutation that is U. U is abnormal base. How might it have formed? Because of some chemical agents that might cause deamination of thymine, resulting in uracil. Uracil is an abnormal base within DNA. It's a base that is found predominantly in RNA and not in DNA. And to release or repair such damage, you will have uracil glycosylases, which will break the bond, glycosidic bond. By the repair mechanism is base excision repair. That is what would have happened if there is deamination of thymine. Then you will have the formation of uracil. So we will get rid of it. I am now going to say a different scenario where you can assume that deamination of thymine is happening and deamination of cytosine, methyl cytosine is also happening. What will you get then? A, and the second base G, and here is, that is U, and this is a methylated cytosine. So deamination of methylated cytosine will result in T. And then you have A, C. A is there, C is not methylated as I said, and then U. What is abnormal base within this DNA and what is not? You can, this is normal, this is normal, this is abnormal. Uracil is not found in DNA, so you should get rid of it, as we discussed in this case. What about thymine? Thymine is a natural base in DNA. And how can the repair mechanisms recognize this as a mutation? And if replication happens, the thymine will just get established because there will be a delay, more delay in removing those thymines. The thymine here is a deaminated methyl cytosine. Actual position is actually in that sequence, it should have been C, cytosine. But because it is a methylated cytosine and deamination of it resulted in thymine. Whereas in the T, here if you had deamination of thymine, it would result in uracil and they would be repaired. I hope you understood why GCs are hotspots for mutations. Okay. So there are several consequences to what happens because of DNA methylation. So typically a gene that is expressed, these are the enhancers, and to the enhancers are activators, then the mediator complex, RNA polymerase, and transcription is active. But what may happen occasionally is the DNA might get methylated. Not every DNA is methylated generally. There will be DNA methyltransferases which will methylate the cytosines. Mostly they target the CpG islands that are nearby the promoter elements and so on. The proteins that we are talking, these proteins, the activators, or the proteins of the RNA polymerase, they are specific to DNA. They are not specific, or they are not evolved as methylated. They cannot bind to methylated DNA. Of course, there are some other proteins that can bind specifically to methylated DNA. But majority of the transcriptional machinery, they only can bind to normal DNA, not methylated DNA. So what happens in this case? Because of methylation, the transcription is off, and they cannot be turned on unless this methylation is removed. And there are also special proteins that can bind to methylated DNA. They coat over this DNA, and then it is furthermore turned off. Kind of, there is no possibility of expression, not even the basal level. And then you can have several other proteins which will play. The histone deacetylases, they will, when the DNA binding proteins, methylated DNA binding proteins come, because what we are showing here is as a linear DNA, but actually they are present as nucleosomes, right? They are also nucleosomes. And the proteins that bind here, they attract histone deacylases and chromatin remodeling complexes. As a result, the what is spread out can, because of histone deacetylation, the chromatin could become much more compressed like this, like formation of heterochromatin. So it doesn't mean that this is the only way. At least I hope you will be able to see how DNA methylation can cause, you're making a chemical modification to DNA, but it is resulting in a structural modification of the whole chromatin itself. In that area, it's not the whole chromosome will get silenced. That's not the case. Okay, so I'll just quickly repeat. Methylation of DNA will prevent the binding of general transcription factors or other transcriptional machinery, including that of the activator proteins, and because of which the gene transcription reduces or goes down. And that will also have an attract the methylated DNA binding proteins, which will further recruit histone deacylases and chromatin remodeling complexes that will result in formation of heterochromatin, where gene expression in that part of the chromosome is completely off. So this I have, yeah. So there is something called as genomic imprinting. These are all so nice to, or amazing in a way. We will see how much we can understand at this stage, but otherwise we will see in the next semester. We all are biparental, or I should say, our cells are diploid, most of the cells. And that's because we are biparental, which would mean that we have a father, and we have a mother. They give us one set each. That means we have 2n, n plus n. One is coming from father, and the other set is coming from the mother. And there are sometimes, say for example, there is a gene A. I'll put it as small a and small a. Like I said in dosage compensation, what did I say in dosage compensation? We do not want both the X chromosomes transcriptionally active, because that could cause problems when we only want one dosage of X chromosome genes. Say for example, I'm saying about, there is a gene A. We do not want expression from this much concentration. That is too much of product of from gene A, because of which it will become complex or complications. We only want either one of it expressed and the other one not expressed. So we have two sets, one from father and one from mother. And in that case, maybe A of this is paternal because it's father, you say, it's not, it's repressed. And from mother, it can be, or the maternal allele, A allele, can be expressed. If there are several genes within eukaryotes, especially in higher organisms, where you will find this kind of arrangement. Some genes are always expressed from the maternal genes, maternal alleles. And some genes are expressed only from paternal alleles. Right? And that kind of expression or regulation is referred to as genomic imprinting. It says allele specific gene expression. Okay, allele specific gene expression that actually depends on the sex of the parent. What I meant is whether it is maternal allele that is expressed, or whether it is paternal allele is expressed. I give the benefit of doubt to you that if this could become a little more, and I'm not going to bother you with asking about the actual core concepts of it. But I would like this to, I would like you to have it as an opening to be ready to be to accept even more astonishing ways. We might not have known about this genomic imprinting before, and when we read for the first time, it feels too weird too. And why should we have something like this unless except to bother us? But this is biology, and these concepts are included in the syllabus to give you an introduction rather than to that you would understand the whole of it. But I will just give you what happens in the general ways. But otherwise, if you know the basic definition of genomic imprinting at this stage, it should be sufficient. It is allele specific gene expression, okay, and that is dependent on the sex of the parent from which that particular allele is derived. So how is this repression occurs, or how is expression allowed, and so on? Usually that expression, the repression of a gene is by DNA methylation. Not limited to, but majority of these cases it is DNA methylation. So this is a cycle, right? What I meant is, say this is an organism. It has two sets. Here is gene A. In A, it is expressed from the blue one, but not expressed from the orange one. You can think of this black marked one as methylation. In gene B, here there is expression, and from the blue one, there is no expression. When this individual is making gametes, right? When that individual making gametes, the intermediate step in some, at the very early embryonic stages, you call it as primordial germ cells, which give rise to gametes. In that case, all the methylation is erased. There is no more methylation. All the gray boxes are done, removed. And then new imprints are made. So if this is a female, you will have, sorry, a male, you will have male type imprints made. If it is a female, then it will have a male type, female type imprints made. I will try to discuss this again, or I will not probably. I can share some of my videos from stem cell biology just in case you would want to know about it. And then the mature gamete. So this gamete, assume that it has come from another individual. Okay, the same individual will not produce both sperm and egg unless it is a hermaphrodite. So if you take mammals, there are no hermaphrodites. You will have parents that are uniparental. I mean, they have unisex or monosex. So they produce gametes, and these gametes will fuse upon fertilization to produce zygote, and the zygote will give rise to blastocyst with the inner cell mass and the trophoectoderm. And say in one case you have gene B is repressed, and in this case gene A is repressed. So there will be an expression of gene A from here, and gene B is expressed from this allele. So you only have one copy expression from A alleles and one from B. There is a balance. This is called as genomic imprinting. Okay. I'll just touch one final concept that we have discussed. This will be the last slide, maybe, and then I have other things we'll see in the next class. Here is genomic imprinting and the expression of IGF2 and H19. Their role and other things, their involvement in splicing and other things I'm not going to discuss. But the important point is about enhancer. There is a imprint control region, or that is an insulator actually. And you have the activator that is bind to an enhancer can activate either H19 expression or IGF2. I told you before in the previous slides where we saw One enhancer bound activators can activate either this or this one, or you can also have one promoter activated by either of the enhancers. So in this particular example here, we are seeing H19, the genetic organization of H19, IGF2, and the imprint control region, or it's a type of insulator. So what happens here is one of it is from maternal and one of it is from paternal. Okay, this is the maternal chromosome, and this is what happens on paternal chromosome. On paternal chromosome, you have methylation of this whole region. If this is okay, before going, I'll talk about maternal chromosome. In maternal chromosome, there is an insulator to which CTCF binds. If CTCF binds, the enhancer cannot reach the promoter elements of IGF2. Therefore, IGF2 is not expressed, but the enhancer can activate a transcription from H19. So you have one dose of H19 expressed. The same or the similar genetic architecture that is present in paternal chromosomes, same IGF2 gene and H19 gene, and ICR also an enhancer. The enhancer is bound by activators, but there is one difference, and the difference is the methylation of this region. If this ICR is methylated, then the CTCF cannot bind here, because CTCF can only bind to unmethylated DNA. And it is sequence specific binding, of course. And because of this methylation, CTCF cannot bind here. So there is no insulation, because of which the enhancer can bend and bind to bend and activate the transcription from IGF2. So if you see here, one another point why H19, there could be two reasons why H19 is not expressed. First of all, the enhancer is not meeting here. And secondly, the promoter element is also methylated, because of which you will not have RNA polymerase and others binding here, or the transcriptional factors. So the enhancer will activate IGF2. So you will have IGF2 expressed, but no H19. So how many doses of IGF2 do we have? IGF2 will be one dose. Although there are two copies of genes for IGF2 and also for H19, only one is expressing the IGF2, that is the paternal allele, and IGF2 is expressed only from H19 is expressed from the maternal allele, right? So this is a little bit complex, but in eukaryotes you will find so many other things. There are also many RNA induced gene regulation where the nucleotide sequence is not changing, but then you end up having changes in the gene expression, and that is because of these epigenetic mechanisms. It could be DNA methylation, histone modifications, or nuclear architecture, chromatin binding, chromatin architecture, and so on. So we'll discuss in the next class, we will discuss about the regulatory RNAs. Again, here also I don't want that you have to study the whole details of it, but have a good introduction to it so that it will be useful in your future courses. Okay, I will stop here, and if there are any questions or anything I would want to discuss.